Advancement in Neuroscience, Unveiling Alzheimer's Disease through Multiphoton Microscopy.
Assalamualaikum warahmatullahi wabarakatuh, and hi, my name is Syahmina Rifqah Binti Saipullah, and I will explain about the introduction part. The definition of the neurodegenerative disease is because it consists of the progressive cell death and brain function decline. It is also caused by various factors like unhealthy lifestyle, genetics, and also aging. This disease have the key mechanism, which is it's disrupting the neurons such as beta amyloid protein deposit disrupting neuron, and also misfolded tau protein disrupting the neuron function, and also the chronic brain inflammation damaging neuron, and then the oxidative stress is when the imbalance between free radicals and antioxidant harming the cell. And then there are some example for the neurodegenerative illness like Alzheimer's disease, AD, and also Parkinson's disease, and so on. The importance of the neuroscience is to understand mechanism for better treatment, diagnosis, and also prevention at the earlier stage. And also we can use the imaging technique like PET and MRI for visual brain changes and to analyze genetic components and neurotransmitter functions. Next, we can see the comparison between the traditional versus multiphoton microscopy, which is for traditional method, it have limited focus and sensitivity to early changes and poor in biomarker detection. But for multiphoton microscopy, there are some few advantages like high resolution, deep penetration, non-invasive, and also we can view in 3D image and real-time visualization. The benefits of this multiphoton microscopy is mapping brain structure at high spatial density and microscopic level, and also we can study about beta amyloid plaques, tau tangles, and cell vascular interaction, and then it generates in 3D image for observe cell movement and synaptic exchange. And last one, it's provide valuable information for developing treatments and diagnosis.
Hi, I'm Fatihah Binti Bakar will present for the part Principle of Multiphoton Microscopy. Firstly, Fundamental Principle. Fundamental principle involve multiphoton excitation where multiple photons interact simultaneously, leading to the emission of a single shorter wavelength photon. It requires high excitation intensity achieved through ultra-short pulse infrared laser and high numerical aperture objective lens. It is result in highly localized excitation within the focal volume of the objective. For the traditional microscopy limitation, it relies only single photon excitation and linear imaging technique. It causes photobleaching and phototoxic effect due to continuous exposure to high intensity excitation light. Limited imaging depth due to scattering and absorption of shorter wavelength excitation light. For the advantage of the multiphoton microscopy, it relies on two-photon excitation and nonlinear fluorescence imaging. It enables real-time observation of individual cells and molecules within intact tissue. It offers extensive depth penetration with reduced phototoxicity, making it suitable for non-invasive imaging. For the imaging technique, address the need for real-time observation of cell and subcellular components without harming the specimen. It allows penetration to depth for several hundred micrometers without physical sample cutting. It uses longer wavelength, reducing phototoxicity and enable prolonged observation period compared to traditional microscopy. For the disease detection, it enables early stage detection of disease and provide enhanced understanding of associated pathophysiological process. It allows characterization of morphological features and cellular function in native tissue. For the biomedical research advancement, it provides high-resolution 3D imaging for unparalleled detail. It offers dynamic imaging capabilities for both in vivo and in vitro high-speed observation. For the optical sectioning and depth penetration, it enables optical sectioning within thick biological specimen without the need for the confocal pinhole aperture, and also increase signal to noise ratio and gather more emission photon. And depth penetration in biological tissue is enhanced by near-infrared excitation wavelength with reduced scattering, and reduced photobleaching and phototoxicity due to highly localized stimulation and a shift from ultraviolet to near-infrared spectrum. Thank you.
Hi, my name is Chun. Today I'm going to present the application of multiphoton microscopy in Alzheimer's disease. In 2001, Christie et al. Utilized in vivo multiphoton microscopy to study theophylline stained synapse in an Alzheimer's disease mouse model. The results suggested plaque stability over time, supporting a dynamic feedback model with real instances of growth or shrinkage and the emergence of new plaques. In Chun et al.'s 2002 research, multiphoton microscopy played a crucial role in vivo imaging of amyloid deposit in transgenic mouse using the amyloid imaging agent metoxid X04. The study highlighted the agent's superior brain penetration, its ability to generate high-resolution fluorescent images of individual plaques and cerebrovascular amyloid, and its potential as a positron emission tomography (PET) amyloid imaging agent for in vivo human study, particularly when labeled with carbon-11. In 2003, McClellan et al. Utilized multiphoton microscopy to investigate the correlation between amyloid plaques and the reactive oxygen species ROS production in AD. The finding revealed a direct link between amyloid plaques and the generation of free radicals, particularly localized around dense core plaques, and suggested that administering the free radical spin trap and tetrabutylphenylnitron (PBN) could significantly reduce the oxidation of photogenetic probes, implying a potential therapeutic strategy for AD by neutralizing free radicals associated with amyloid plaques. In the 2005 study by Spire et al., gene transfer technique were used to introduce green fluorescent protein GFP into neuron in an Alzheimer's disease transgenic mouse model. Multiphoton imaging of GFP-labeled neuron exposed disrupted neural trajectories and reduced dendritic spine density near amyloid plaques, highlighting significant synaptic integrity loss and altered neuronal structure associated with dense core plaque in Alzheimer's disease. In the 2019 study by Jawaharian et al., deep convolutional neural network DeepWest were utilized to automate the segmentation of 3D in vivo multiphoton images of vasculature in mouse model, specifically focusing on Alzheimer's disease. DeepWest demonstrated superior segmentation accuracy over state-of-art method and human annotators, reducing manual annotation time significantly and revealing insight into minimal differences in capillary diameter and tortuosity between young and old mouse model of AD, with a decrease in longer capillary segment in aged animals. In 2020, Cavo et al. Utilized multiphoton microscopy in a transgenic mouse model to examine the influence of amyloid beta on mitochondrial calcium level in Alzheimer's disease. Their result indicated that amyloid beta aggregate lead to an elevation in mitochondrial calcium, potentially causing neuronal cell death and blocking the mitochondrial calcium uniporter with RU360, show promise as a therapeutic approach to mitigate this effect in Alzheimer's disease.
Hello, my name is Nurul Binti Ahmad Shukri, and I will explain to you the challenge of multiphoton microscopy. First of all, it is limited imaging depth. The scattering of light in brain tissues limits the penetration depth, making it difficult to visualize structures in regions such as the hippocampus and cortex. The second one is resolution constraint. High resolution imaging across large tissue volumes is time consuming, limiting the study of intricate neural circuits and cellular dynamics. The third one is fluorescence labeling requirements, which is dependency on fluorescence label may introduce toxicity and hinder specific cells type targeting. The last one is data analysis complexity, which is managing large dataset requires advanced tools for segmentation, tracking, and interpreting of dynamic neural networks. This is an advancement and innovations for multiphoton microscopy. The first point is advanced optics and imaging techniques, which is adaptive optics and novel light source improve penetration depth and image quality in deep brain regions. The second one is enhanced resolution methods, which is integration of super resolution techniques like state microscopy and SIM for finer scale studies. The third one is non-linear imaging modalities, which is integrated for label-free non-linear imaging modalities such as THG and CARS. The fourth one is Machine learning for data analysis. Development of machine learning algorithm for automated image analysis, segmentation, and feature extractions. The lastly is in vivo imaging techniques. It is miniaturized imaging probes and techniques for in vivo imaging for deep brain structures.
There are few limitations in this multiphoton microscopy. First, cost and equipment complexity. Multiphoton microscopy requires expensive equipment including high-power laser and specialized optics. Second, field of view constraint. The limited field of view compared to other imaging techniques potentially hindering the ability to comprehensive imaging, which is large brain area or complex neural network. Next is depth limitation. Despite excelling at deep tissue imaging, multiphoton microscopy has restricted penetration depth. In conclusion, multiphoton microscopy significantly advances Alzheimer's research by enabling detailed, real-time observation and analysis of amyloid deposit and related cellular processes. This technology offers valuable insight into early-stage identification and monitoring of Alzheimer's disease. By providing high-detail imaging, dynamic observation, and deep tissue investigation, multiphoton microscopy serves as a fundamental in biomedical research for understanding complex biological processes in Alzheimer's. This immediate application contributes to the development of specific therapy options and hold promise for future breakthrough in both the detection and treatment of neurodegenerative disorders.